We're going to start today with two very excellent researchers in cereal biology, physiology and genetics, with Jesse Poland here in KAUST and Michael Dingkuhn in CIRAD. We're going to kick off with Jesse. It's fantastic to have both of these gentlemen here. Jesse has joined KAUST relatively recently, come from Kansas State University, and here in KAUST he's established or is establishing, I should really more correctly say, his professorship in plant breeding and genetics with his laboratory. He's calling the Plant Breeding and Genetics Laboratory, where he's going to focus on developing climate resilient crops, as many of us are in fact doing. Jesse really has he really is one of the best plant breeders in the public sector in the world. He really is an extraordinarily talented young guy, and it's fantastic to have him here, and great to have him as the first speaker in this series. As an indication of his skills, besides his amazing publication citation record, is that he leads a USAID program, Feed the Future Innovation Lab for Applied Wheat Genomics, a multinational gathering of people which are dedicated really to improving sustainable yield increases and yield stabilities in a range of developing countries. So with no further ado, I'd like to introduce Jesse to introduce some of his research and the possibilities of collaboration with CIRAD. Jesse.
Thanks, Mark. You can hear me okay, and hopefully see the slides now. Looks okay? Great.
Yes.
Okay, thanks, Mark. It's too generous of an introduction. And so, yeah, glad to join for everyone. 9 years, it'll go for 10 years total, to implement genomic selection in the CIMMYT wheat program. And so I just wanted to kind of take a stroll down memory lane, so to say, and to give everybody an idea kind of where we've come from and what things are going on. And so this is a nice aerial picture of one of the field testing sites in India. This is a really large project with the CIMMYT main program in Mexico, and then across South Asia, India, Pakistan, Bangladesh, Nepal. And so it's been a really great project, many collaborators, just so many great colleagues, too many to mention. So how do I go forward here? So just by way of introduction, I always like to give an idea of the world of wheat and where we're looking. This is a heat map showing where wheat's produced around the world. Like I noted, focus here in North India and Pakistan, major wheat growing area, breadbasket, supporting hundreds of millions of smallholder farmers, and huge implications for food security. And that was something noted by Dr. Borlaug when he was getting the Nobel Peace Prize, that the food security and global peace are really go hand in hand. So the scope of this is that there's an increasing demand for wheat every year, going up by tens of %. And given if we just leave our current varieties on the current production practices, you'd actually have a decreasing from new diseases, climate change. And so in a plant breeding perspective, we need about a 2% gain per year. And the current estimates from historical studies are around 1%. So we need to do a lot more breeding and do it a lot faster. So what this looks like projecting, here's the historical yield trends. This is a little outdated to 2010, but you can see that this projection, the projected demand is actually an increase from the current rates, and that's getting pushed down, especially by climate change and diseases. And so we need to push this up with better agronomics, and then we're really focused on how do we increase and develop improved varieties and do that faster and more efficiently. So really to focus, the breeding cycle is this iterative process of making crosses between, usually between elite varieties and then through a long evaluation process, selecting for things like disease resistance, agronomics, in later stages, selecting for yield, and then in final stages, also selecting for quality. And so obviously this evaluation process takes a long time, and it's also quite expensive, especially you get to these later stages. A yield plot can be in the order of $10 to $50 per plot, and a quality baking test on the order of $100 or hundreds of dollars. And so to really make this faster and more efficient, we think about a lot about different breeding methodologies. So this is a lot of quantitative genetics framework behind it. And one of the equations to think about here is this genetic gain over time. And so it's a function of the selection intensity. So how big the populations are and what percentage is being selected, the accuracy. So this is obviously how accurate your phenotyping, how close that how close that is to the genetic value, and then the genetic variance, so the diversity of the population. And then that's an inverse proportion to the years per cycle. So the faster you can make this cycle go, the better the genetic gain over a function of time will be. So plant breeding then really turns into a numbers game. And these are actual numbers that come from the CIMMYT main program. So the program there is running bulk populations and actually literally looking at the early generations, F2, F3, looking at millions of individual plants in the field, making selected bulk populations, up to about the F4, F5 generation, where they derive around 40 to 50 or 60,000 single plant derived, and those go into small plots, which are about half a meter by half a meter in size. And those are selected out and go to preliminary yield trials, 10,000, and advanced trials, and then the international nurseries. And so to keep these numbers in mind, this is the point we'll come back to when we're implementing genomics is on these orders of tens of thousands. So thinking again on this breeding cycle, there's a lot of approaches both on the phenotyping optimization to make this run bigger and faster and more efficient. And so one of the things that was just stepping back now and want to walk through kind of this trajectory of how we came to implementing genomic selection, just to give a historical perspective, this is a really foundational paper in 2001 by Mewison et al. And this prediction of total genetic value using dense genome-wide markers. So basically genome-wide markers to predict the breeding value. And their conclusion from this was a simulation paper because it was so far ahead of its time. Their conclusion here was that this will make these dense marker maps. They could see that the molecular markers were developing very quickly. So rather than having tens of markers, we were quickly getting hundreds or thousands of markers. And this would make genotyping many individuals feasible. And then they concluded, based on these completely new models that they had developed, that you could predict the total genetic value, just like you would predict from classical breeding evaluations. And so this was really a game changer, but it was so far ahead of its time that it took about 10 years for this to really get rolling in any type of practical application. And so to give you an idea how far this was ahead of its time, this has been cited almost 5,000 times as of this year. And I don't have the numbers going all the way back, but 5 years after publication, it still only had 13 citations. So this paper had an impact factor of like almost zero, even though it is probably one of the most trajectory changing, transformative approaches to hit the plant breeding world, hit the animal and plant breeding in the past few decades. So what is this genomic selection? Like I mentioned, it's a prediction of the total genetic value, and this is using genome-wide markers. And so essentially we're estimating the kinship or the realized relationships. And then from that, you can predict the phenotype. So this is very familiar to us. If you think about twins, they have the exact same genetics and they're typically very similar. The difference is if you think about siblings, they share some genetics, but at the same time, there's variation. And then if you think about just a group of diverse random kids, there's many different phenotypes. And so the real key is to use the molecular markers in the essence of differentiating siblings, because by pedigree, they all have the same relationship, even though they're genetically different. So for implementing genomic selection, there's two real key things that have to happen. And this is again, going back to 2009, when we were really starting to think about putting this into practice, is having a training population. This is to train the model where you have genotypes and phenotypes, so you can basically estimate the genetic values. And then from there, you can implement that on selection candidates where they're only genotyped, predict the phenotype, and make selections. The real key thing, though, here is that you have to have inexpensive, high-density genotypes. And at the time, we had for like wheat, there was just starting to come along, maybe some array-based methods, but those were in the order of hundreds of dollars per sample. And you think about if you're going to do 1,000 or 10,000 candidates, and from there, you're going to basically throw away 90% of them. That gets to become a very expensive exercise. And then the other thing is to have accurate phenotypes. But in the course of the breeding program, there's already really extensive yield testing and phenotyping going on. And so the real key missing piece here was this idea that we needed high density genotypes that were cheap enough that we could do huge populations. And so this came around then at kind of this introduction in 2007, 2008 of next generation sequencing technologies, where we were able to generate huge amounts of sequencing data, getting exponentially cheaper. And so with the development of genotyping by sequencing, we could leverage these amazing developments in sequencing output. And at the same time, we could get this really low sample cost, which was absolutely the key for implementing this in actual breeding program. The interesting part is, as I was looking back at some old things, and this presentation, this came from 2013, thinking about at the time, like, right, this next generation sequencing was really a paradigm shift for everybody. And there's really a lot of questions if this could be useful at all, especially for something like genotyping and doing breeding and selections and stuff. So we were part of these developments. We got these working really well. We could like do them on biparental populations and things like that. So the GBS is everyone's quite familiar with it now. But at the time, there was really a lot of questions whether this would work, how this would work, if we could use this for actual genetic studies. And so one of the first things we did was to do some cross-validation studies, and that's these cross-validation accuracies, where we basically take genotype of population. This was just a few hundred individuals, and then you basically leave out 10% of the population, build the model, and then try and predict that other 10%. And so what we could do with the GBS markers at the time was we compared to this DART array, which was sort of the state-of-the-art standard at the time for genotyping wheat populations, that we could actually show that the GBS markers were working as well or even better than the DART markers for this cross-validation accuracy. So we could get prediction accuracies up at 0.3 or 0.4 for yield. This might not seem really great, but given the heritability, these are actually quite good predictions. And so this was kind of good empirical theoretical evidence that maybe this could really be used as the that key piece of a cheap genotyping tool that we needed. And so we then went for, and this USAID project got initiated in 2013, and really with the scope of implementing genomic selection at the full scale of the CIMMYT program. And so what then was sort of obvious actually looked like then is that we could use those genomic predictions to predict things like rust and diseases. We could predict yield, we could predict quality, and we could actually make selections early on before going through these long evaluations and make that cycle go much, much faster. So the point that I want to really bring to today and think about in agriculture innovation is there's a proof of concept and there's some nice paper that comes out, but then there's an additional piece to really make impact that goes way beyond that. And so that's why I was thinking about this saying of the last mile and or the last 1.8 kilometers if you're in metric. So The idea being that in the telecommunications and things, they would run these main, or electricity, they would run the main lines on the main route. But then the last mile was actually connecting the individual houses to this telecommunications network. And that last mile is always the most difficult, the most expensive, and it's definitely the most expensive relative to the number of households being served, because it's running it only to one household. And so in this agriculture innovation and developing new breeding methodologies, proof of concept, but then getting that implemented in the actual breeding program at scale is really that last 10% that takes 90% of the effort. So just to give an idea of what this is starting to look like as we worked extensively with the program implementing this for wheat quality here. So here back again to the CIMMYT bread wheat program. This was just the first 5 years that we did this, but you can see the number of entries that are being evaluated for yield on the order of 8,000 to 10,000 a year. Just a few thousand of these are evaluated for quality because it's much more laborious and expensive. And then there is, and subsets of these were evaluated all over South Asia. So since the initiation in 2012-13, we've actually been genotyping 10,000 a year using this GBS for everything that enters into the yield trials to build these big prediction models. So in the area of quality, we have things like 1,000 kernel weight, protein hardness on the grain, all the way down to milling and baking properties like loaf volume. So you can think about one of these loaves of bread being a single data point. And then we're actually going to build a genomic prediction model to predict that, which obviously is going to be much faster and much cheaper than baking a loaf of bread. And so we were able to do this once these data sets got into the thousands of samples and individuals. Our predictions actually become quite good. So here you can see predicted versus observed. Loaf volume, we have a good correlation. And the prediction accuracy of these is 0.5 up to 0.7, which based on the heritability and based on the accuracy of the phenotypes, this is actually exceptionally good. So back again to our genetics theory, we're able to estimate then the expected gain from this indirect selection, because as we implement this in the program, you can't actually observe as you move forward, because it's just too extensive to basically replicate an entire breeding program and thus have like a case control kind of study. So with this, we have notes that we're phenotyping about 2,000, but we can genotype 10,000. And if 1,000 of those are advanced, then we have a much higher selection intensity from genotyping and genomic prediction. And we also have the heritability for each one of these quality traits. So from that, we can estimate the relative gain. And here again, this is largely driven by the much larger population size. We haven't even yet factored in the ability to move the cycles faster. And so you can see here the heritability of these traits ranging from 0.4 up to 0.6, and then the correlation between the observed and the predicted. And then that gives us this relative response to selection. And anything that's over 1 is actually going to be favorable for the genomic prediction with a larger population size. And so we got quite an extensive, up to a doubling of the expected gain by using genomics implementing for quality. And so this has really been extensively implemented in the program for the past 5 years since its initial work, and predicting quality for all of these traits in the CIMMYT program, and then using that in the selection for advancement. I just want to highlight quickly in these last 2 slides before we stop is that we can also, it's not just a black box approach that I explained at first, but essentially we have a huge population that's genotyped with high density markers. And so we have a ready-made association panel with extreme amount of power. And so just to highlight here a couple cases, this has been used extensively just to better understand the germplasm, the genetics, what genes are controlling these traits. So for example, here's resistance to wheat blast. There's a major allele on chromosome 2A at this translocation that shows up here in this association for the mean evaluation of wheat blast severity. And then also these are breeding lines being haplotypes for each of these QTLs. And you can see versus the green versus the blue, the breeding lines are running vertically here. There's thousands of them here. Now we're able to take a haplotype approach also and make some informed decisions on which kind of parents to be crossing and how to optimize combinations in the crossing block to really keep moving this forward. So it's not a completely black box that we can start to open this up and look at optimized breeding strategies. Just to give more ideas, this has been utilizing all of these large data sets. Here showing days to heading for some major vernalization and other PPD for relative maturity, and then also hitting known stem rust genes and all of the different evaluations. And so just to conclude, the key to all of this was being able, down in this bottom left panel, this is showing the genome-wide marker density, being able to generate these tens of thousands of markers for less than $10 a sample was really that key piece that then we could do this at scale on tens of thousands of breeding lines every year. So that's kind of a quick walk down memory lane, stuff that's been going on over the past 10 years. Like I mentioned, there's been a phenomenal group in my group, but then also, especially with Ravi Singh and all of the breeding group at CIMMYT, just wonderful times traveling together across South Asia, working together, dreaming up new breeding strategies, and slowly, convincing Ravi that, this genomic stuff would really work. And as Mark mentioned at the start, this was largely supported by USAID and also the Gates Foundation. And so here's me, as we were going down memory lane, here's myself as a graduate student. I got to meet Dr. Borlaug for a moment years ago before he passed away. And so he said, go for plant breeding and give it a good push. So I'll stop there, and that's what we're trying to do. So as we make these good innovations, they need to be pushed through the last mile. So good. How do I stop? Stop.
Great. That's
okay.
Fantastic.
Thanks, Mark.
Very impressive. Thank you very much, Jesse. I'll never look at those telegraph poles in the same way again. I love the way they're all kinky and wobbly.
That was, I don't know if that's from Kansas, but that's how they look in Kansas.
Yeah, no, that's right. They look in a lot of the world. Good. All right then. Are there questions for Jesse about his wheat breeding, genomic selection, genetic markers? I was wondering about how you account. This is a very naive question, so be gentle with me, Jesse. But G by E, how does that? Yeah, okay.
I didn't even touch on that, but that's a huge question. In some ways, it's the same problem with genomic prediction. In some ways, it's different. On one hand, there's been a lot of work going on, especially stuff led by Jose Crosa at CIMMYT, because we have all these multi-location trials, right? And even if they're not balanced, meaning the same breeding lines in the same years at different locations, with the genomics, you can essentially replicate alleles across the locations. And so there's a lot of work going on to develop better G by E prediction models. And they do, so the crux is they do really well if you know what environment you're trying to predict. However, there's always next year when you don't know what the environment's going to be and you can't predict it well. So that's one hand. On the other hand is that because we're able to basically use date, so rather than evaluating the line per se, and you only get to evaluate that in however many environments you plant it, you can essentially use in the entirety of the historical data to get a better assessment, even though you only evaluate them in limited environments. So as far as selection for yield stability and yield across environments, the genomics are actually quite powerful for that.
Yeah, that's great. Thank you very much.
May I ask a question?
Are there other? Yes, Michael. Excellent.
Yeah. Just quickly, very impressive. I love it. But as you're in KAUST now, could you just say a few words about how KAUST is involved in breeding, contributes to breeding, or how do you envision that? Because I guess in Jeddah you're not going to do all that, but you've been trying.
Yeah. Well, I mean, there's a couple answers to that one. So on one hand, I mean, this was, we were leading and working on this from Kansas extensively. And there's a lot of wheat in Kansas, but it's not Simmet spring wheat. So this entire project, even though we were working, was really this phenomenal international collaboration. So, I mean, that's one real key first thing, right? Is that we're tied into these networks, and the breeding is really a network. So that's one thing. The other thing that's really struck me since being here is there's a lot of, I don't want to say like low-hanging fruit, but there's a lot of breeding innovation and breeding work that needs to happen on other sort of orphan crops and crops that just don't have breeding work going on. I mean, I'll throw date palm out there just because it's a multi-billion-dollar crop in the kingdom, and there's virtually no breeding efforts. And so this is something we really need to push forward. And that's just, for date palm, that's not even here. I've been getting, more background, Egypt, other places. These are huge, huge crops that are just very minimal or no breeding efforts at all. And so you think about like so much of our breeding work goes into the major three, right? Wheat and corn and rice. But now we have the methodologies. We can put a genome together overnight. And so I think we have huge opportunity space to take what we've learned in the big ones and put it to a whole bunch of other species.
Okay.
That's good. Yeah, that's a nice question. Bulo Debeza, I don't know if you want to read out your own question or elaborate on it, or leave me to read it out. There's a question in the chat box.
Yeah, I see I don't know.
So it's actually partly what I've already covered.
Yeah.
My great minds think alike. How do we include G by E and mass micro-assisted selection in genomic selection prediction? How many locations is needed? I mean, how long is a piece?
So that's always a magical question. It really depends on, how correlated are the locations from year to year and between each other. So we've actually been doing a lot of this extensively on the South Asia locations. And you find locations that are very well correlated to each other all the time, right? So they share a lot of information. And you have other locations, other sites, other years that they're not correlated to anything. So obviously with those, you need to evaluate in the representative locations. So there's no magic number. It really depends on how variable the locations and the years are. And from that, right, if they're all the same, you just need one location, right? If they're all different, you need to go for all of them.
Yeah, sure. Actually, I'd like about your second to last slide. You had a trait for which you had a strong QTO, DTHD or something.
Sorry, I didn't say that's like days to heading, relative maturity.
Oh, days to heading, of course. Yeah, okay, right here. Okay, and then
I just wanted to give a few snapshots that all of these traits that are measured. We essentially have a population size of 10 to 50,000. So you can map QTLs for everything.
Not quite everything, Jesse, but lots of things, sure.
Everything that we measure.
Yeah, that's right. Good. Are there any other questions from the broader audience? I could ask Jesse questions all day, but I think that would be mean and selfish. Good. All right then. Thank you very much, Jesse. Really appreciate that. Let's move on to Michael. So Michael is another, I think, fabulously thoughtful, deeply thoughtful researcher whose work I've admired, and who's, as a person, I've admired for a long time. He's German, now lives in France, but he's worked for most of his life in the tropics, in the wet tropics in the Philippines and in West Africa, in particular on rice, but on more than just, in inverted commas, rice. And he works on yield and stress physiology using phenotyping and modeling. He's got a very nice mathematical mind and applies this very well, very appropriately to plant physiological issues. He was important, very important, essentially important in the global rice phenotyping network. In fact, I think he founded this, and he and I think that's how we met actually in the rice phenotyping network. But he works on a range of things related to, of course, phenotype genotype associations and effects of CO2 on crop performance. And on that note, I think I'm just going to be quiet and leave the floor to Michael, and just be very glad that you're here, and thank you for joining us, Michael.
Thanks so much, Mark. I have to just see how technically this works. What do I do first? I share my screen, right?
Yes, that should be almost enough.
I select my screen, which is probably this one.
And then hit share screen again. That's it.
Yeah.
And then you go into presentation mode.
Now I make it big.
That's right. So hit the presentation button. That's it.
And
You're
then
in business.
We can go.
That's it. Yep.
Great. So I was asked to talk about research on cereal crop improvement at CIRAD. And I'm adding a subtitle, some innovative research questions. Well, I'm not a breeder, and I'm a physiologist and modeler who works with breeders and has been working with breeders most of his life. But I cannot really talk competently about breeding. So I will talk about traits of interest, and it's in contrast to the previous presentation, I'm offering something like a basket of ideas. That is my approach here. So my topics are specific challenges, but today, how we see it at CIRAD, cereal crop improvement happening at CIRAD, what is that? Then I will touch briefly on 4 case studies from our own kitchen here that might provide ideas for collaboration, and I will conclude on those. So what are today's challenges? I don't think I should run into open doors here. How do you call this? It's a German expression. It is, of course, in large part about climate change and environment change. And on the left-hand side, you see this shocking spike of CO2 in the atmosphere that is really, compared to the past 20,000 years, or 10,000 years, if we take the time when crops were grown already, but this is a thing never seen. And then we have a whole range here. Do you see my cursor? Of climate and climate change related constraints that affect the crop. And we have, of course, the demand side for the crop produce. On the other side, that is the field in which we are situating ourselves, just like so many others too. Now, what is concretely happening at CIRAD? Now, in general terms, we do breeding in partnership, generally. There's very little autonomous breeding happening at our centers. We work with NARS, with the CG institutes, particularly CIAD, Africa Rice, ERI, EQUISAT, with academia, of course, a lot, and KAUST is of great interest to us in that way, and with seed companies. We expatriate experts, such as breeders, geneticists, or physiologists, to partner locations for several years, sometimes also shorter, sometimes very long. That is how our collaboration works. CIRAD as a center, as a physical center, is a pre-breeding hub also. There's a lot of breeding support research happening in terms of genetics, diversity, genetic diversity, phenotyping, GWAS, G/E studies, and modeling of all that. In terms of cereals, we work on the improvement of rice and sorghum mainly these days. Maize has been dropped quite a while ago. And I think INRA has taken that mandate. We work on sugarcane, which is of course not a cereal, but genetically and physiologically not so distant. And there are some orphan crops, but not really serious breeding going on them right now. We work on the tropics and the Mediterranean temperate environments. To be more specific, what are the traits or activities in breeding, starting with rice? There is here in Camargue in France, breeding work on salinity, so salt tolerance. We work on rice heat and cold tolerance in various locations. There is a major breeding program in Madagascar that is run in large part by CIRAD. We work on drought and acidity and aluminum toxicity tolerance, mainly in South America with partners there. And there's a lot of work, of course, on biotic stresses, particularly fungus diseases in many places. In sorghum, we work on grain and forage quality. With the private sector, we work on seedling cold tolerance, on heat and cold tolerance at enthesis, which is quite a different thing again, with the question of pollen viability and things like that. On drought in Senegal and with the US, there's a collaboration. And in more general terms, with many partners and many projects on sustainable intensification, which is something like a global motto of our research, on forage and grain yield and quality, and on phenology, that is acting with that, of course, on climate resilience, pests, and name it. Now I just want to mention a couple of issues that I cannot see properly here because your pictures are masking it, but I will try to figure it out. Ongoing research that is relevant to breeding. In rice, we work on traits for improving yield potential and yield stability. We're working on heat avoidance by transpiration cooling of different organs of the plant, on traits for lodging resistance, storm proofing, if you like, of plants. We just started working on agri-photovoltaics, shade tolerance of rice and possibly other crops in the future is they are grown under solar panels on a commercial scale. In sorghum, there is currently working on priming potential of repetitive stresses. So in a way, maybe epigenetic or conditioning of physiological stress responses. In rice and sorghum, we work on the role and the diversity of the root parenchyma and how that relates to both too much water and too little water, and there are maybe trade-offs there. And finally, in rice and wheat, we are working on the adaptation of those plants to increasing CO2 levels and how they are able to better use these in the future. So I will now run you quickly through four thematic trade types, let's say, that we are interested in and that might be of interest for collaboration. The first one being, can we boost CO2 response of C3 crops to achieve greater yield? And as you all know, C3 crops show a very strong response in terms of yield or growth to CO2 levels. And that is a slide here that I got from Paul Quick, where they just lined up plants treated with different levels. You can see that there is a current level, there's a pre-industrial level highlighted here. 2050 is a sort of landmark. And So these are the levels of changes that we are anticipating or observing. Interestingly, in phase trials, CO2 trials in the field, in this case here in Japan on the right side, it was observed that yields would be increased by between, let's say, 10 and 30%, depending on the variety, when CO2 was increased by 200 ppm. That is quite a large range. And this differential responses of varieties to CO2 can be related to their sink capacity. And that brings me to our motto here. If you apply an additional carbon source in the form of CO2, you must have a plant that can use it. It has to have the sinks. This is not only a matter of using assimilates to fill grains, it is something happening throughout the plant cycle. You see on this slide a study that we did together with IRI and Japan. We looked at the compensatory phenotypic plasticity in terms of tillering potential and how that relates to yield responses under enhanced CO2. And the model system was here to plant plants, genotypes, two big populations at different spacings, and giving them an opportunity to fill those spaces through more or less tillering, which is then an expression of their plasticity of sinks. And actually the QTLs that you get through GVALs from those then gave us significant signals, and for a couple of QTLs, we grew in the CO2 phase in the field, accessions having the minor or major allele. And in fact, this trait did translate into increased CO2 responsive yield. At the level of the leaf, Denis Fabre has done some very interesting work very recently. He manipulated source and sink through CO2 levels and through panicle pruning in rice. And among the things that he discovered was that, okay, when sinks become limiting, you have a decrease in photosynthetic rate. You see that here. And that's happening in the afternoon, not in the morning. So actually you have to look at the whole day, kinetics of photosynthesis, especially look at the afternoon to see what is really happening quantitatively. Now, from this, he deducted that if I take plants, genotypes that naturally or constitutively differ in source-sink ratio, and there's a simple trait combination identified here. It's called the local source-sink ratio. If you do that, you measure that here on 5 genotypes that are contrasting their response to CO2. Here we have the Amax photosynthesis 800 over 400 ppm. That response can be positive or even negative. Here we have, on the right-hand side, we have insufficient sinks to absorb the CO2. You get feedback inhibition of photosynthesis. And there are plants that have a high sink compared to the source, and those have a phenomenal response to CO2 in terms of yield, in terms of photosynthesis, and so forth. You can simulate these things, and we don't have the perfect model on that, but the Samara model was able to capture that to some extent. And we can see here that for the grain yield response simulated, to atmospheric CO2. We have two curves here, one for a low tillering and one for a high tillering variety. It's the same variety, and we just manipulated the tillering parameter. And you can see that makes a hell of a difference whether you're able to tiller in response to CO2 or not. It is mainly biomass and yield that are affected. The plant height and even the LRI are not very much affected. We can see here on the third graph, that actually the increase from CO2 of yields from pre-industrial to today's levels are much higher than actually what we can expect for the next 20 or 30 years. And so actually, theoretically, our yields have very much benefited already from increasing CO2, but maybe other things have been counteracting that. At any rate, the take-home message is here on the right-hand side that a lot of photosynthetic potential remains unused if we don't have plastic sinks that can use that photosynthetic. And the question is, how can we stimulate things to make crops more carbon hungry? I will not go into this here now, but just as an outlook, we would be really interested in collaborating on genes and alleles that can stimulate zinc activity in the plant in different organs. And the T6P feast and famine system that, for example, Matthew Paul in UK is working on a lot. That is an extremely promising candidate because it is something that works across all higher plants and is operating in all organs and at all developmental stages. There's a family of genes, and these are really central to tuning plants in the way we are suggesting. So this is a potential target for collaboration. Now, quickly, another set of traits: avoiding heat stress via transpiration cooling. And I had a PhD student, Cecil Julia, who did thousands and thousands of thermal images in the field in Senegal, Camargue, and in France and in the Philippines also, panicle temperature and the microclimate and so forth. And we saw that even though Senegal had the hottest climate, maximum temperatures here for that particular period, the panicle temperature was not all that high, whereas in the Philippines, the panicle temperature was higher than that of the ambient. So there is a strong climate interaction in terms of the cooling, and panicles are warmest in the tropical humid climate and not so much in the very hot desert environment. This can be easily explained, of course, with VPD, vapor pressure deficit of the atmosphere, and we have a linear relationship between the cooling effect, temperature differences, a difference between the organ and the air, and on X here, the VPD. And we have the panicles. The panicle up here and the leaf down here. There's a difference between those two, but both operate in a similar way. No, I go to the next slide for lack of time. What is interesting for those four varieties that we tested or studied in this way, is that this thermoregulation is extremely effective as long as VPD is high enough in the atmosphere. When the air temperature goes to 40 degrees or so in real time, the leaf temperature will just level off at, below 30 degrees actually. We have a cooling effect of 12 degrees or so. And so these leaves will stay way below the critical temperature for photosynthesis. And there are lots of models that are crop models that go wrong there because they don't take this into account. The panicle, it is not as extreme as that, but the panicle also has a temperature that levels off at very high ambient temperature. And panicles can largely escape heat-induced sterility. Actually, in this table, we can see between the two yellow bars the range of conditions, VPD and air temperature, that occur together. The hotter it is, the higher VPD normally is anyway also. And so that is something that we find extremely interesting. Future research should address the question of genetic diversity. Can this be bred for? But then also, what are the trade-offs with water use? What are the effects of water deficit? Because transpiration cooling is evidently water consuming. Third set of traits we are interested in can be storm-proof rice and other crops for that matter. Work I conducted with Suchit Shrestha from Nepal at Erie, and that was funded by Syngenta. Now, we developed what we call the rice blaster, which is no rocket science, but extremely useful with the, sorry, what's happening here? It's just an electric turbine and the electricity generator. And why is it doing that? And we achieve up to 60 kilometers per hour wind speed over a distance of 6 to 8 meters in the field, which is shown here as an air channel in the field. And with that, we can very reproducibly induce lodging and study it, compare the observed lodging with various traits, see how much recovery there is from lodging. There is none actually, and so forth. So we have 20 varieties here, where the actual lodging is well correlated with what is called a lodging resistance index. That is just an aggregation of traits that contribute to lodging resistance. And the whole thing was validated in the field with natural lodging occurring through storms and high nitrogen inputs that chosen to make the plants a bit more willing to lodge. So we were able to validate this method. I will not spend time on this now. We have lots and lots of trait correlations, and I just want to highlight that stay green traits and sweet stem traits and things like this have quite an effect on lodging resistance. This can, of course, also be simulated and be adapted to the Samara model to do just that. And we explain only about 60% of genotypic differences in lodging with the model, but this model only takes into account simulated height, biomass, stay green traits, and stem thickness, not taking into account chemical traits that we measured but did not simulate, such as lignin, sugars, well, I mean, all these cell wall components, for example, silica too. And very last topic I want to bring up is the issue of making use of multi-location phenomics coming from network research. So if you have populations such as our prey, indica, rice panel, 300 accessions that has been grown in many, many environments, and for some of these environments, the same traits were observed, and we have databases there that are totally underused because there was simply no time before the project ended to really analyze everything. Looking into yield potential and yield stability traits, we have here 10 environments that include Asian and South American and African environments. And we just plotted the genotypic grain yield against the population mean grain yield, which gives you some sort of stress index that breeders use. And you have, for example, in green here, a variety that has a high yield potential, but at the same, but a low yield stability. You have in red R64, a mega variety that has a fairly good yield potential, but also very good stability. Then there are those, of course, that have low potential but high stability. And from that, we derived here a yield stability index and a yield potential index. Of course, yield potential and stability are negatively correlated, but not in a very strict way. So there is room for compromise varieties or varieties that achieve both to some extent. And we have now lots of data from GWAS that we are playing with. And I can't go any further in this here now because that's where we are. We have traits like days to flowering, plant grain yield, grain weight, harvest index, panicle number, spike number, fertility ratio, and things like this for the 10 environments. And the color indicates here the significance of a certain QTL. These are 15 different QTLs. You can see that there are many traits, many QTLs that are highly pleiotropic, sometimes for just physiological reasons. Others, like this is ST1 that sticks out like a sore thumb. Of course, the semi-dwarf breeding gene. Yeah, I finished basically. We want to try to relate the allelic diversity on those QTLs to the stability and potential parameters of yield that we have obtained across all those sites. So my conclusions are as follows, and I focus on ideas for KAUST CIRAD collaboration. From our 4 case studies to develop breeding approaches for enhanced CO2 adaptation, transform this cognitive research into breeding tools and phenotyping tools. It's actually going on. Extend this to other C3 crops, other cereals, or quinoa, to be discussed if there's interest. Transpiration, cooling, do we have genetic diversity? Can we breed for it? How does it interact with water deficit? Or if that be the case dramatically, then should this only be of interest for irrigated environments? Breeding and modeling of lodging resistance, storm proofing of crops, and the question what makes a mega variety, how to explain and then predict, maybe also genomically predict yield potential and yield stability. Furthermore, and what I have not been going into now is obviously soil tolerance is of interest to us and to you as well. Drought and heat tolerance traits as well. The root and parenchyma is a very interesting subject that has potential of adapting crops to other hydrologies. And finally, the 4P, 4 per 1,000 context, the initiative to bring more carbon into the soil through root carbon accumulation, and particularly lignins that stay in the soil for a long time, can benefit from elevated atmospheric CO2 to sequester more carbon in the soil in different plant types. So that is what I wanted to say. Thank you very much.
Excellent. Thank you very much. That's really very, very good. And thank you for entering into the spirit of proactively exploring the collaborative possibilities. That's much appreciated, Michael. Thank you.
That's how I understood.
No, that's great. No, that's really, really good. Thank you. Yeah, I mean, we can do that. We can also sort of strut our stuff and then let people cherry pick. But you are more proactive, which is good. Both approaches are valid. Thank you very much. So there are questions for Michael that people would like to ask. Aha, we have Branda Wolf.
Thank you. I have a question with regards to growing your plants under elevated CO2 concentrations. Did you see any acceleration of the generation time under these conditions? Do they flower earlier and set seed earlier?
Not significantly, no. I would expect a slight accelerating effect through slightly increased plant temperature, because they transpire less under higher CO2, so they are warmer. But we have not seen that effect really. I think in Japan it has been seen and has been published also.
It would depend on other limiting factors as well, the warmth.
Of course, but if you just take CO2 per se.
Okay, thank you.
Yes, but it depends on how warm. Warmer isn't necessarily better.
No. On the contrary, actually, higher CO2 increases the risk of heat-induced sterility and other heat-related negative effects.
Yes. Actually, while we're talking about heat, I mean, I found your panicle temperature effects and comparison between West Africa and the Philippines particularly interesting, and it resonated immediately with me in a way I must admit I hadn't thought of before, which is with the date palms. So when I first arrived here, it was blisteringly hot, and I was cycling around this place, and there's these date palms sitting up there all day in the massive heat, and I was thinking how incredibly impressive they were. And then I started to thought, yeah, but they're using a huge amount of water each day, right, and transpiration and cooling. Yeah. But it can be really humid here in the summer, just when it's also really hot. And there'll still be a very, much reduced vapor pressure deficit, even though the temperatures are high, because the humidity is really high. So maybe I should be thinking again, reappraising the impressiveness of date palms and their heat tolerance. Because I must admit, I thought, well, you're just sweating a lot, but maybe they're not. I think that could be, I mean, date palms are an incredibly impressive plant because of their temperature tolerance.
Your strategy has to depend on the resources that you have on site. And if you have a lot of water, if you're near a big river, an estuary or something like that, there is no harm in using a lot of water.
No.
In the middle of the desert, I would probably argue differently.
Yeah, but date palms need to be irrigated. They require a lot of water.
Yeah.
But it was the humidity layering on top, which I hadn't thought of, that making their transpiration cooling approach more difficult. So that was pretty interesting. I also thought it was very interesting your comment, your conclusions about the benefit of plastic carbon sinks. The plants being able to respond to the future increased CO2 by having, well, in your example, more tillers, but
not only
other things. No, exactly. There'll be other
roots
ways.
as
well. Yes, absolutely. Yes, especially in the context of your CO2 sequestration, the soil project. Yeah, that's very good. Are there any other questions from anybody else at this stage? Or do we need to think? I think your talk was full of ideas. I got a whole page of notes. So maybe people are needing time to assimilate and take on board some of what you were saying. By the way,
question on chat box. One question.
Where do I find that? Let me see.
Okay, so Bulo again, with a fertile mind and much thinking. Thank you. He's saying, how could you avoid heat stress through transporting cooling temperature to crops? So Bulo, you're thinking about the bulk flow of water to the shoot, and that water being cooling down. I'm not sure if that would be quantitatively significant compared to latent heat from evaporation. Can you clarify that, Michael?
I mean, I've never thought about putting blowers into the field and having evaporators maybe and blowing cool air from those evaporators into the field. You do that in greenhouses, right? But it sounds expensive and less elegant to me than what the plant is actually doing already.
I'm not sure that's what Bulo meant. I was thinking, well, I'm not exactly sure. I don't know if Bulo wants to come and join this conversation, but I think he was thinking about the movement of the cooled water from the soil, from the bulk volume of the soil.
Okay.
Shoot, I'm not sure.
No.
Anyway, okay.
I mean, irrigation water is used for the opposite frequently in rice and in vegetables to prevent cold stress and freezing.
At night, yes.
Like that.
Yes.
Yeah. Or to have more inertia in the system. One can think about this in terms of heat also. Would be discussion.
Yeah, I mean, I have thought about it in greenhouses where you can cool the deep water culture for leafy greens, for example, which causes a locally lower temperature around the base of the stem, at least, which is reducing bolting to flowering in leafy greens.
Yes, in the Rosetta plants,
that's just a tiny little lettuce.
The meristems are down at the bottom. It's
exactly. Yeah. Vanessa.
Hi, Michael. I just have a question about. I was also interested in your transpiration cooling of panicles. Obviously, we're interested in heat stress responses of quinoa, and I'm wondering if you've come up with any great way to measure panicle temperature in the field, that being in a high throughput manner.
Good question.
No, basically, you have two ways. One is remote by infrared photography or scanning. And the other one is thermocouples, which is sort of a little bit more tedious. And I don't see many other ways to do it. But obviously, infrared photography is a fantastic tool. There are some problems you have to deal with, and you have to do it from different sides.
Yes.
You have to consider where the sun comes from and whatever. But
also the panicle architecture can be quite a problem.
Pardon?
The panicle architecture can be quite a problem with this imaging.
I mean, they're very good cameras, and you can go down to the smallest detail, even on a photography that captures 100 panicles in the field, and you can see the stem, the anthers. You can see you see the temperature of all those. All depends on your equipment.
Wow.
All depends on your equipment. You have to be a bit careful also with errors coming in the instrument itself from the instrument temperature.
Yeah.
But these are all things that can be solved, and this is the way to go.
Okay. For rice, yes. Yeah.
Not only. No.
No?
No, no, no. Why? Why?
Oh, it'd be great if we could apply it to quinoa, which has high variability in panicle architecture, height, all these sorts of issues with imaging.
It's all a matter of image processing afterwards.
Okay. Interesting. Yeah. Thank you for your answer there.
That's encouraging. Yes, thank you. Rod Wing, Professor Wing.
Don't hear you.
You're still on mute. I think you're still on mute, Rod. That's one of the buttons. Oh, no, sorry, he's done it.
I'm not that old. I liked your talk, Michael. Very, very exciting. And so this is a more of a logistical type question. And in the beginning of your talk, you mentioned that CIRAD has a program of stationing scientists remotely. And I know you're a big benefactor of that, as was Erie. And how does that program work? And also, does CIRAD accept scientists, let's say from KAUST for a couple of years? I'm just exploring ideas like that, because I think having a really strong relationship with CIRAD is very appealing and very exciting for us. And when a scientist comes from CIRAD, is it primarily for research, or can it be for research and teaching?
It can absolutely be for research and teaching. It should not be for teaching alone.
Yeah.
But research and teaching, CIRAD has a mandate also for capacity building for partners. Now, KAUST is evidently a bit of an off type when it comes to tropical partners, because you are sort of part of the rich world, or you are another rich world. But it is appealing to CIRAD, certainly, to work with places like you. And expatriation is of scientists for 2 years, 3 years, 5 years, and some people don't come back. That is something that is at the core of the way we function. That said, in the not so distant past, this was easy to do. There was budget for it, and the scientists were not forced to find every cent to pay for what it costs. Now, financially, this is a matter that needs negotiation, and CIRAD is not doing things for free anymore, and things like this. But absolutely, CIRAD is interested in that, and CIRAD is not a for-profit company. We are a company, actually, public.
Okay. Yeah.
But we are not for profit. But then we have to sustain ourselves also. So you can see what direction that goes. The same applies to hosting scientists from partner institutions. And we are very much interested in getting high-level scientists that advance our, our scientific progress. Just as we are interested in scientists from poor countries in order to strengthen their home institutions, to which we hope they will go back afterwards.
Okay, so it goes both ways, which is good. I mean, that's because I would say KAUST is very unique, but we're definitely a well-financed university, but I would say the rest of the country has limitations in terms of agriculture and their expertise. And the Center for Desert Agriculture, only has 10 faculty. You guys have probably hundreds of PIs with some expertise that we don't have. So I think tapping into that would be very beneficial for us and for expanding the mission of sustainable agriculture in Saudi, which is something that we're really pushing. So
we followed up, absolutely.
Yeah.
And perhaps also in third-party countries of mutual interest, you know.
Yeah.
Like,
I heard that KAUST is involved. Oh, sorry. KAUST is involved in research in Africa, in many places in the world.
Yeah.
And we can have triangular collaborations too.
Yeah, exactly.
Yeah.
Exactly.
Okay, thank you, Michael, and look forward to the rest of the seminar series that you guys have put together. It's exciting.
Thanks.
Good. And on that note, that's perfect. Thank you very much, Rod. Thank you, Michael, and thank you, Jesse. And I think on that note, with no further ado, I should just ask you all to join me in thanking both speakers, and thank you all for some good questions and conversation as well. Cheers. Thank you all.
Thanks for inviting.